So we've been interested in complex coacervates as model prebiotic compartments because they are very easy to assemble, and they can accumulate different biomolecules that are very relevant to prebiotic chemistry. So a little bit of background on how these guys form is through assembly of polyelectrolytes. So I'm showing here polyanion and polycation. They can assemble together to form these dense phase and dilute phase. So the dense phase actually contains a lot of polyelectrolytes and charged molecules. Since RNA is highly charged, they can also be expected to accumulate within these droplets. So before I joined Phil's lab, a graduate student in the lab, Erica Frankel, she had done a lot of nice work with the system of polyallylamine hydrochloride and ATP, where obviously the polyallylamine is the polycation and the ATP is the polyanion. And when you add these things together, you form these really nice droplets that accumulate. I'm not going to go into too much detail, but what she showed was you could really accumulate magnesium, nucleotide, and RNA. For example, when she used a DP and polyallylamine to form these coacervates and only added about 5 millimolar magnesium bulk, the concentration of magnesium in the coacervates were upwards of molar. So that's tremendous enhancements of magnesium levels, which might be very relevant for chemistries like non-enzymatic RNA polymerization or even ribozyme catalyzed RNA polymerization as well. Okay. And obviously what she also found was that different RNAs were also highly enriched in these coacervates. So what we started to do was we started exploring different polycations and polyanions to form these coacervates and ask whether they can accumulate RNAs and whether they can participate in chemistries that we care about in terms of RNA world hypothesis. So you can see that oligolysine, polydienyl dimethyl ammonium chloride, PDAC, olyarginine, they all form these nice coacervates when we add oligo-RNA to the solution. And what I didn't tell you was that we actually added Cy5 or Cy3 labeled RNA in these coacervates, and you can see that the RNA really accumulates in these droplets. And we're going to use the same RNA for template-directed RNA polymerization that I'm going to talk to you guys here. So again, these are the different coacervate compositions that we're using. We're using poly-A-RNA to have the polyanionic part, and we're using polyallylamine, PDAC, oligolysine, oligoarginine as the polycationic part. And what we do is we add this RNA primer template complex, and you can see that the template has a stretch of C's where this activated nucleotide, that is G, can come in and the primer gets extended. And what we do is that we add everything together and then we separate the two phases so that the coacervate phase is separate from the dilute phase, and we monitor the reaction in these two phases separately. So what happens? When you don't have any coacervates, of course, you see that the RNA starts to, the primer starts to get longer because this monomer gets incorporated. And what was really interesting was that in the case of the dilute phase, which is the supernatant phase, you don't really see that much RNA, which is expected because a lot of the RNA ends up in the condensed phase inside the coacervates. But what was striking was that in the condensed phase, only the coacervates that were formed from the PDAC and oligoRNA, they supported the non-enzymatic polymerization. And you can see that all the ones that don't support non-enzymatic polymerization with PAH, oligoarginine, and oligolysine, they can not only engage in charge interactions, but they also have this ability to hydrogen bond. So we think that this super high concentration of hydrogen bond donors, acceptors, and charge groups within the coacervates, they might be somehow inhibiting this reaction, potentially misfolding or unfolding the primer template complex. Since we knew that some of the other coacervate systems increase the magnesium concentrations within the condensed phase, we thought it might be interesting to look at non-enzymatic polymerization in different magnesium concentrations in the presence of coacervates. So you can see that without any coacervates in buffer, as you decrease the magnesium level, you do take a hit in how long the RNA is formed by this reaction. And what was really interesting was when we did the same thing, but in the presence of PDAC and oligoRNA coacervates, you do take a little bit of it in the beginning, but then even when we didn't add any magnesium, you still formed like decent amount of +1, and you even see some amount of +2 band formed from non-enzymatic polymerization, and that's quantified here. And so to really get into more detail of what was going on, we decided to add an excess of EDTA to the reaction. So when we added EDTA, a very little background reaction that was happening even in the absence of magnesium, that goes away as well, as expected, because there might be some background divalence that the EDTA is chelating. And what was really surprising was that when we added the PDAC, which is the cationic polymer that we were using to form the coacervates, we actually rescue the reaction back. So what this was telling us was that not only that coacervates can act as this passive compartments that are just taking up RNA molecules, but they might be actually engaging in the chemistry as well. So that was really interesting. So having looked at non-enzymatic polymerization, we decided to now go study a little more complex system, and the sequel to this you've already heard yesterday from Phil, where we wanted to look at ribozyme catalysis within complex coacervate system. And the model we used is the hammerhead ribozyme, which is shown here. So the black strand is the enzyme strand, which base pairs with the substrate strand, which is shown in green, and then cuts the substrate right where this red triangle is. And so the idea was, if in an early Earth, the enzymes were probably not very abundant. They were probably very scarce. And so for the enzyme, for this particular reaction to happen, of course, the enzyme needs to find the substrate. So if you are, and by the way, this is just a native gel that shows the association between the enzyme and the substrate. So if you have a lot of enzyme present, then you start to associate with the substrate, so the molecule becomes larger, so it moves up. But if you don't have enough enzyme, you're below the dissociation constant, then you don't see much association, and so the substrate is not really interacting with the enzyme. And presumably, when the functional molecules were very scarce in the early Earth, we were probably dealing with this sort of regime. So can complex coacervates activate these kind of chemistries when you have very limited functional RNA molecules? So let's see what happens. So again, we tested these sort of ribozyme reactions with PDAC and oligoaspartic acid coacervates. So the way we do these experiments is we first add the polycation and the polyanion to form the coacervates, and then we add the ribozyme and the substrate separately and monitor the reaction. So to our surprise, indeed, well, in case of buffer where we don't have any coacervates, you don't see a lot of reaction, which is the cleavage of the substrate, because of course, since there's only 5 nanomolar enzyme and the KD, I think at 1 millimolar, we estimated it to be about 250 nanomolar. So there's not a lot of association between the ribozyme and the substrate, so you don't see a lot of reaction. Whereas when we have the PDAC and the D10 coacervates, you see robust enhancement of the ribozyme chemistry. So what we think is going on is that the coacervates are bringing these RNA molecules together and promoting RNA-RNA interactions that are not possible in dilute solutions. We tested several different coacervate compositions. For example, PDAC and oligoaspartic acid, obviously they activate the ribozyme. Oligolysine and oligoaspartic acid, these coacervates also tended to activate the ribozyme. Oligoarginine, on the other hand, we saw inhibition of the reaction, and we do think, and I think based on Irene's work, we know that arginine is highly interactive with RNA, and I think the oligoarginine is probably misfolding the ribozyme when it gets inside the complex coacervates. And finally, what we decided to do was we decided to test whether this sort of system is widespread. Can other enzymes also be activated using this mechanism? And so this is the 1023 DNAzyme, which is from Jerry Joyce's lab, and it also catalyzes similar chemistry. The DNA strand base pairs with a substrate, which is shown in blue, and cuts where the red triangle is. And here you can see that with high enzyme, the DNAzyme concentration, you can see nice cleavage of the substrate, but when you reduce the DNAzyme to 5 nanomolar, you don't see a lot of chemistry being happening. And when we do the same reaction now in the presence of complex coacervates, you can see that the DNAzyme also gets turned on. And we have done this with also hairpin ribozyme, which I didn't have time to talk about. In that case, the enzyme actually gets activated by concentration of spermine, which is not really an RNA, but a small molecule. So there are multiple ways that coacervates seem to be enhancing different nucleic acid enzymes. And that's just the quantification from different magnesium levels. So in summary, what I want to talk about is that, we've shown that specific complex coacervates can not only partition RNA molecules, but they can also enhance template-directed polymerization under limiting magnesium concentrations. And concentration of RNA and other co-solids can lead to enhancements of RNA catalysis. And finally, we've shown that complex coacervate-mediated enhancements seem to be general, as in different composition of coacervates can activate different nucleic acid enzymes. So we think that this might be a very useful thing to study for diverse nucleic acid enzymes. And with that, I would like to thank Phil's lab and Chris's lab, who's been really instrumental in, and we have had an awesome collaboration in all those liquid phase separated systems. And we're starting to look at liquid phase separation in cells with Geraldine Sidhu at Johns Hopkins. We're starting to do some ribozyme catalyzed RNA polymerization with Jerry Joyce, and we're also looking non-enzymatic oligonucleotide phosphorylation with Ramkrishna Murthy's lab. So a lot of cool things going on. I'll be happy to take any questions.